Okay, let's continue. So last time we talked about the introduction and also pairwise sequence comparison. In the lecture 3, I will talk about multiple sequence alignment and the phylogenetic tree. So here is about this example of a multiple sequence alignment. So in the multiple sequence alignment, all the sequences are handled equally. Then you align them at the corresponding positions. Then what you see that some position, the amino acids are fully conserved. For example, the cysteine here is fully conserved. And then some other position fully conserved. But then you also see some positions, they are not fully conserved, but here highlighted in yellow, they are negatively charged. So E and D, so they are both one negative charge. And then other parts you see gaps. So here it basically gives you a snapshot of evolution. That is, these proteins have evolutionary relationships, even though in multiple sequence alignment, you do not see which one came first, which one came later, but at least you see which part is highly conserved. So why we need to do a multiple sequence alignment? Obviously, it is a natural extension of a pairwise sequence comparison. So we talk about this two sequences, we can just use alignment algorithm to put them together. We also talk about this side blast, that is, you can use one sequence to align many, but that's not a multiple sequence alignment. That is using one to check others, but others, they do not align with each other. So you can get a so-called sequence profile from side blast, but you won't be able to get a true multiple sequence alignment. So true multiple sequence alignment is extremely powerful. So I really like this quote by Tim Hubbard and others. That is, the pairwise alignment whispers, multiple alignment shout out aloud. That is, with multiple sequence alignment, you see very rich information. You can make it to do a lot of predictions, to study a lot of interesting biology. And it is much more sensitive in terms of sequence relationships than pairwise sequence comparison. And also multiple sequence alignment is a foundation for other bioinformatics methods. For example, how to construct a phylogenetic tree. It is pretty much built on top of a multiple sequence alignment. In fact, phylogenetic tree construction and the multiple sequence alignment, they are called a dual problem. That is, in order to solve one problem in an optimum fashion, you need to solve the other problem. So that's why they are closely related. In fact, you will see at the algorithm level, typically people do both things simultaneously. And then multiple alignment is the foundation for protein family classification. So through multiple sequence alignment, you see those sequences with evolutionary relationships. You can further divide them into, let's say, superfamily, family, and smaller groups. And here is an example of a multiple sequence alignment. So let's see how we can utilize information. I just give some very high-level example. So here, this is the result of a cluster W, cluster omega, actually. We'll use a very simple example, which I took from this link. There are 3 RES proteins. And then when you align them, first of all, you see the format. This is a standard cluster format. You have a gene name here, and then each line represents this one sequence. And then the corresponding position are aligned here. If they are the same amino acid at a position, you put a star here. If they are related amino acids, for example, T and S, threonine and serine, they are related. They have similar biochemical properties, and they have the same phosphorylation properties. In fact, many cases, the same phosphorylation, the same kinase can phosphorylate both threonine and serine. So you put this colon here, and then for other places, you just put this empty space. That means that they are not that related. You could put just one dot here. That is, they are somewhat related, but not as closely related as I show here between T and S. And then the color, of course, is not part of a cluster output format, but it is for annotation purpose. You see the green portion of the protein, they are 100% identical. So that means that in evolution, this part of a protein sequence is highly conserved. So that typically means that they are functionally highly critical. If you mutate any of the amino acids in this block, it may change the function dramatically. So that's why it is highly conserved. Then other parts you see more or less conserved. And then you also see here towards the end, there are more variable regions. But actually, the issue that how we define this variable region, if I do it, I probably would do it maybe differently. I probably would go all the way at least till 170 or a little bit more, because at 170, you still see 3K. And then 171, L and I, they are also highly similar. So at least I can go up to 171. But this is a little bit subjective. The point is that I typically read the alignment in more detail because I have done many of the protein modelings in collaboration with biologists. I found that using this alone probably not good enough. You really need to see a little bit more details. I just give one example. In fact, there are many things you can see. One example you can see here that at the position 151, KRAS, you have R, which is positively charged. And you have two Gs in other two proteins. So that means that this is, of course, highly disruptive from this glycine to a positively charged residue. So the point is that whether they would change the function entirely, then you need to look at the bigger context. Unfortunately, this bigger context cannot be handled by current alignment algorithm very well, because the current alignment algorithm is just do column by column. But if you see a little bit more biology, you see some interesting pattern. That is here, the third one, KRAS, you have a positive charge, aspartic acid, arginine. And then, but you see the second protein, NRAS, you have a positive charge here, which is histidine in 131 position. But then the first protein, it also have a extra positive charge somewhere, that is histidine here. So even though at each position you see dramatic change, but the charge somehow is balanced. In fact, if you look at the protein structure, it's balanced even better. So the point is the alignment, there are a lot of things you can see. This kind of knowledge is not reflected in algorithm, but if you do this kind of a modeling more and more, then you can accumulate such knowledge. So I just give a simple example. Next, I'd like to talk about phylogeny tree or phylogenetic tree. So we have these sequences in multiple sequence alignments, and then you can see the conservation pattern, etc. But if you like to see in a more detailed structure, then you need to build a tree structure. And this is so-called a phylogenetic tree. In the phylogenetic tree, you have these nodes at the end of the tree, and each node represents a sequence. You can use the species name of that sequence to represent a node. And then these sequences will be connected. Here, the number shows the sequence identity between these two sequences. And then you can build the hierarchical relationships. And then the number here shows the average sequence identity between this group and this group. And then you can build the overall tree like this. So multiple sequence alignment and the finite genetic tree, they are so-called a dual problem. That is, if you want to solve one rigorously, you need to solve the other one rigorously at the same time. In practice, people often do this iteratively. For example, you could build a simple multiple sequence alignment and then to form a guided tree, and then you can use the guided tree to help you do more rigorous alignment. So this kind of a problem is actually so-called NP hard. That is, it won't be easy to solve in a so-called fast time rigorously. So that is, you have to use an approximate method to solve this problem. And so when you see the results, you need to take a grain of salt, because these results, they are heuristic results. It doesn't mean they can really do the alignment or tree construction in a global optimal fashion or in a rigorous fashion. There are actually two types of trees. One is called a species tree, one is called a gene tree. A species tree basically means how these species are related. In this case, typically you will choose one sequence as a representative for that species. And then basically you have 10 species, you have 10 sequences. And these 10 sequences, if they are aligned, they're supposed to be so-called orthologs. That is, you can experimentally replace one gene in one species by the ortholog gene in another species, and the function doesn't change. Of course, this is a very high bar to define the orthologs. Computationally, you can find methods to predict orthologs. And then species tree, you can really see when speciation took place and all those speciation events. The other type of tree is a so-called gene tree. That is how the genes are related. In that case, you can have several genes from one species. You can have several species mixed together. In this case, it can reveal some gene duplication event, some species event, but it requires you to do more annotation in order to understand the relationships among these genes. Here, I want to use one example to demonstrate the power of a multiple sequence alignment and a phylogenetic tree. This is called phylogenetic footprinting. This is a study of some genes in Arabidopsis. It is in collaboration with Professor J. Seelan at our university. So we did some sequence alignment and also see the changes in the phylogenetic tree. This figure shows the sequence identity between any two genes in this group. So in the bigger group, all these 5 genes, they are similar in sequence identity. But there are 2 groups. One is a BCCP group. We have 2 genes, BCCP1 and BCCP2. The other group is called the BADC group. We have 3, 1, 2, 3. And then here shows the sequence identity between any 2 genes. For example, here is the 51% between BCCP1 and BCCP2. And then the structures of these genes, they're all very similar. But if you look at the sequence alignment, you do see some interesting patterns. So BCCP1 and 2, they have a conserved pattern here. And in BADC group, this pattern is lost. But then in BADC group, you see an emerging new pattern. So this probably suggests some function in BCCP1 got lost, but some new function is acquired in BADC1. So you see that the sequence alignment can really reveal things like that. Then we studied the phylogenetic footprinting. So the idea is that you use a taxonomy tree. So this tree is not built by us. It's based on taxonomy tree standard. But we can map the genes to each species. And then interestingly, we see that in red algae and early species, we do not see BADC genes. We only see BCCP genes. But then after red algae, like in green algae or more advanced plant species, you start to see BADC genes. Of course, in different species, they have a little bit different distribution. But the big part is that the BADC gene probably emerged after red algae and start to be present in green algae, and later becomes more advanced. You have even more like 3 types. So this kind of tree analysis and multiple sequence alignment can really help you to understand the function and evolution of the underlying genes. If you want to know more about this work, you can look up this paper